Hello everyone, and thank you so much for joining me today. My name is Toshio Inui, CEO of the Seisei Group in Japan. Today, I'm excited to talk to you about dietary MAF, and more specifically, whether immune proteins like MAF can have powerful anti-aging effects, including telomere extension, upregulation of telomerase, TERT and TERC, Klotho gene overexpression. This is a big question, but by the end of this video, you'll understand the answer clearly. Before we begin, let me clarify one important point. MAF was originally described as a macrophage-activating factor. However, recent evidence shows that MAF actually functions more precisely as a macrophage normalizing factor, returning macrophages to a balanced homeostatic state. Let me begin with a quick introduction to Seisei. Our head office and cell processing center are located in Osaka, and we also have clinics in Kobe and Tokyo. Although we have primarily treated cancer patients over the years, today we see patients from all over the world with a wide variety of health conditions. Seisei is now expanding internationally, establishing branches around the world. Our mission is simple but ambitious: to help people overcome serious diseases such as cancer, autism, infectious diseases, and many other conditions that impact human health. By growing globally, we hope to make these treatments available to people everywhere. We currently collaborate with 6 universities in Japan. Interestingly, 4 of them are not medical universities. Why is that? Because to develop innovative cancer and immune therapies, we need expertise from engineering, science, veterinary medicine, and pharmacy. Each university contributes unique strengths that help us develop more advanced and effective treatments. Now let's move on to the core topic: macrophages, GcMAF, and dietary MAF. Seisei holds worldwide patents for both injectable GcMAF and oral dietary MAF. As we discuss anti-aging and telomere extension, you'll see how these immune proteins play a remarkable role. Most people imagine macrophages as hungry white blood cells that engulf bacteria and pathogens, moving like amoebas. In reality, macrophages are present in almost every tissue of the body. Macrophages perform crucial tasks, engulfing pathogens, removing dead or dying cells, clearing senescent cells and cellular debris, supporting adaptive immunity, promoting wound healing, tissue repair, and regeneration. In the video shown here, you can see macrophages extending their membrane arms to engulf foreign substances known as lamellipodia formation. This diagram shows how macrophages are distributed throughout the body, in the brain, skin, lungs, gut, and many other tissues. Tissue-resident macrophages are essential for repairing damaged tissue, regenerating healthy tissue, maintaining overall rejuvenation of organs. Their importance cannot be overstated. GcMAF stands for GC protein-derived macrophage-activating factor. At Seisei, we currently have 5 types of MAF: old GcMAF, new GcMAF, autologous GcMAF, dietary MAF, recombinant MAF. The old version of GcMAF used a vitamin D3 affinity column and carried a higher risk of cross-contamination, so we discontinued it many years ago. Since then, our focus has been on developing new generation MAF treatments. Among them, dietary MAF has shown unexpectedly powerful effects in both animal and human studies. Our latest development, recombinant MAF, could potentially become a new form of medication. So, does dietary MAF really have strong anti-aging benefits? The answer is yes. Dietary MAF is produced using vitamin D binding protein derived from milk. It is classified as a food product in Japan and a dietary supplement internationally. Our research using both mice and human data has revealed remarkable findings. Dietary MAF extends telomere length, upregulates telomerase, TERT and TERC, increases Klotho gene expression in plasma, brain, and kidneys, reduces biological age, improves cognitive function. In mouse studies conducted at Kagoshima University, dietary MAF significantly increased telomere length. It upregulated telomerase, TERT and TERC in both young and aged mice. These results strongly support its potential as an anti-aging therapy. Next, let's look at the scientific evidence behind dietary MAF and its anti-aging benefits. Animal studies show that dietary MAF significantly increases telomere length in both young and aged mice. It also upregulates telomerase, TERT and TERC, which are essential components for maintaining and extending telomeres. These findings suggest that MAF supports cellular longevity at a very fundamental level. This is our newly published article in the journal Scientific Reports. Today I'd like to walk you through the key findings of this paper and explain why they are so important. This figure shows telomere length in young mice after 2 and 4 weeks of treatment with water, MAF, or whey protein. As you can see, 4 weeks of oral MAF intake led to a significant increase in peripheral blood telomere length. Compared with the water group, telomere length increased by 54% after 4 weeks of MAF intake. This figure shows the mRNA expression levels of telomerase reverse transcriptase, TERT, and the telomerase RNA component, TERC, in young mice treated with water, MAF, or whey protein. As you can see, 4 weeks of oral MAF intake significantly increased the mRNA expression of both TERT and TERC in young mice. This figure shows telomere length and the mRNA expression of TERT and TERC in both young and aged mice. As you can see, 4 weeks of oral MAF intake significantly increased telomere length in aged mice, as well as the mRNA expression of TERT and TERC. It almost appears as though the aged mice have reverted to a more youthful state. Next, I'd like to talk about the Klotho gene. So what exactly is the Klotho gene and how does it work? And importantly, how can we boost or supplement Klotho levels in the body? The name Klotho comes from Klotho, one of Zeus's 3 daughters in Greek mythology, the goddess who spins the thread of life. In the legend, Klotho spins the thread, Lachesis measures it, and Atropos cuts it. This symbolism reflects the powerful role the Klotho gene plays in regulating lifespan and aging. This slide illustrates how Klotho functions as a key regulator of aging. As shown here, mice that lack the Klotho gene have an extremely short lifespan, about 80% shorter than normal. The middle bar represents normal mice with typical Klotho levels, and importantly, mice that overexpress the Klotho gene live about 30% longer, so the amount of Klotho in the body has a profound impact on lifespan. Dietary MAF increases the Klotho levels in the plasma, brain, and kidneys of mice. This experiment was conducted by Dr. Katsuura and Dr. Kawamura at Kagoshima University. This figure shows the effects of MAF on plasma Klotho levels in mice. Compared with both the water group and the whey protein group, the MAF group showed a remarkable increase in plasma Klotho levels, almost double. This figure shows the effects of MAF on Klotho levels in different regions of the brain. Compared with both the water group and the whey protein group, the MAF group showed a remarkable increase in Klotho levels in the hypothalamus and amygdala. There was also a modest increase observed in the hippocampus. This figure shows the effects of MAF on Klotho levels in the kidneys of mice. Compared with both the water group and the whey protein group, the MAF group showed a remarkable increase in kidney Klotho content. The kidneys normally contain the highest levels of Klotho in the body, and about 80% of the Klotho found in the bloodstream is produced by the kidneys. We conducted a phase 2 clinical trial in Japan to evaluate the effectiveness of dietary MAF on cognitive function in elderly individuals undergoing rehabilitation. The MAF group showed clear improvements in cognitive scores compared with the control group. Interestingly, many participants in the MAF group did not contract COVID-19 during the study period, which was a very encouraging observation. We conducted a phase 2 clinical trial in Ukraine to evaluate the efficacy of dietary MAF in hospitalized COVID-19 patients. The results were remarkable. We observed a significant reduction in mortality, as well as a rapid recovery from lymphopenia, the decrease in lymphocyte count. This study, led by Dr. Galina, was published in the journal BMC Infectious Diseases. We also conducted a phase 2 clinical trial in Italy to evaluate the efficacy of dietary MAF in hospitalized COVID-19 patients with pneumonia. We saw excellent results there as well. Importantly, no participants in the trial died from COVID-19. During the study, Dr. Lucrezia asked me why participants' kidney function, reflected by eGFR, improved within just 3 weeks of taking MAF triple. At the time, I didn't have a clear answer, but now I believe it's due to the increase in Klotho produced in the kidneys. This study was published by Dr. Lucrezia in the journal Biomedicine and Pharmacotherapy. Dietary MAF. suppresses inflammatory responses induced by lipopolysaccharide in mice. It has strong anti-inflammatory effects, as demonstrated in both mouse experiments and clinical studies. This paper was written by Dr. Katsura and Dr. Kawamura at Kagoshima University. Chronic inflammation is closely linked to aging and cancer. Factors such as oxidative stress, microinflammation, glycation, and smoking are known to accelerate telomere shortening. This study clearly shows that Dietary MAF possesses strong anti-inflammatory properties, which may play an important role in slowing these aging-related processes. Nature Portfolio recently highlighted Saisei Pharma for our breakthrough research in anti-aging immunotherapy. I'm truly honored that our work has been featured in Nature Portfolio once again. 13 years ago, the journal Nature introduced GcMAF as our next-generation immunotherapy. It was a major milestone for us and marked the beginning of our ongoing work in advanced immune-based treatments.
Next, I'd like to introduce our Phase 2 clinical trial conducted in Japan, which evaluates the effectiveness of Dietary MAF on rejuvenation and longevity. I'll walk you through the key findings from this important study. Here is the study design. The primary objective of this Phase 2 trial was to evaluate the effectiveness of Dietary MAF on rejuvenation and longevity. The secondary objectives included measuring telomere length, conducting DNA methylation analysis, including TERT, and analyzing age-related gene expression profiles. The study population consisted of healthy adult men and women over the age of 40. Exclusion criteria included pregnant women, cancer patients, individuals with severe diseases, and those with severe allergies. Here is the study protocol. Participants took 2 capsules of MAF triple per day on an empty stomach, and blood samples were collected at 0, 3, and 6 months. DNA was extracted from peripheral blood, and telomere length was measured using PCR. A total of 203 participants were enrolled as of December 23, 2022. Of these, 139 were female and 64 were male. The age distribution included participants in their 40s, 50s, 60s, 70s, 80s, and even one participant in their 90s. DNA methylation analysis and age-related gene expression profiling are also being conducted as part of this study. This figure shows telomere length at 3 and 6 months. A total of 161 participants completed all measurements. As you can see, telomere length increased steadily over time. Compared with the baseline at 0 months, telomere length increased by an average of 12% at 3 months and 23% at 6 months. These changes are highly significant, with p-values below 0.01. The t-test with equal variance assumptions also confirmed significant differences across all comparisons, again with p-values less than 0.01. This figure shows the gender differences in telomere length between women and men. As expected, women tend to have slightly longer telomeres than men, by about 5%. And as you can see, telomere length increased in both women and men at 3 and 6 months after taking MAF triple. Telomere extension was observed in both genders equally. This figure shows the effects of MF on plasma A Klotho levels in humans. As you can see, plasma A Klotho levels increased by about 20% at both 3 and 6 months after taking MAF triple. This figure shows the changes in telomere length and biological age over time. From the 161 participants who completed telomere measurements, we selected 30 men and 30 women who showed the highest levels of telomere extension for further analysis. This figure shows telomere length at 0 and 6 months. As you can see, telomere length increased dramatically over time. Compared with the baseline at 0 months, telomere length increased by an average of 57% after 6 months. This figure shows biological age at 0 and 6 months. As you can see, biological age decreased clearly over the 6-month period. Since the intake period for MAF triple in this study was only 6 months, I believe this downward trend may continue with longer use. In other words, continued MAF triple supplementation has the potential to further reduce our biological age, helping us stay younger for longer. Here is a summary of our findings. In mice, dietary MAF increased telomere length and upregulated telomerase, both TERT and TERC. It also increased Klotho levels in the plasma, brain, and kidneys. In humans, dietary MAF increased telomere length by an average of 12% at 3 months and 23% at 6 months among 161 participants. It also increased plasma Klotho levels by about 20% at both 3 and 6 months. Furthermore, in the 60 participants who showed the greatest telomere extension, dietary MAF reduced biological age by an average of 1.35 years.